My name is Lora Hooper, and I'm a professor in the Department of Immunology at the University of Texas Southwestern Medical Center at Dallas, and I'm also an investigator of the Howard Hughes Medical Institute. And so in my second talk in this series, I'm going to talk about how we maintain symbiotic host microbial relationships with the microbes that colonize our intestines. So in my first seminar in this series, I talked about how the human intestine is colonized with between 10 and 100 trillion bacteria. And this is in contrast to our own cells, which number only a trillion. So again, we're really at best only 10% human. And I talked also about the fact that these gut microorganisms confer significant metabolic benefits by liberating energy from our diets that we can then take up and utilize. So an enormous question in the field is, a very important question in the field is how we keep these bacteria confined to the lumen of the intestine and prevent their escape into deeper tissues where they can cause infections and disease. And so I'm going to answer this question in part today by talking about some of the research that's ongoing in my lab and that we've carried out over the last 10 years. So the question really is, how do we coexist with 100 trillion bacteria without becoming diseased? So much of the work that's been ongoing in my lab for the last decade or so has focused on the intestinal epithelium. So this is the lining of the intestine, and so this is a schematic of the small intestine of a human or a mouse, depending on what you're looking at. And the main point is that there's a single layer of epithelial cells that lines the entire surface of the intestine. This is really the major interface between your deeper tissues and the trillions of bacteria that inhabit the intestinal lumen and confer these very important metabolic benefits. So the intestinal epithelium is really a critical gatekeeper of the body. And so it's very important for determining the outcome of these host microbial relationships. And it's for this reason that we focused a lot of our efforts on understanding how the epithelium responds to our microbiota. And we've done all of this by doing experiments in mice. So in my last seminar, I discussed briefly the use of germ-free mice as essential experimental tools in microbiota research. And so this is also an essential experimental tool in my lab. And this, again, just shows the basic experimental setup, which consists of a plastic isolator enclosure, the interior of which is sterile, and then we can raise mice under completely microbiologically sterile conditions. So these mice don't have any microorganisms in or on their bodies, so no bacteria and no fungi. And so we can use these mice to investigate how epithelial cells respond when they encounter a bacterium for the first time. And we can use that to learn about how epithelial cells keep these organisms out of our deeper tissues. So one of the approaches that's been really key for this effort is uses a series of techniques that are referred to as transcriptomics. So basically what we've done is to start by comparing the genes that are expressed in germ-free mice, and we determine which genes are expressed using an experimental approach involving DNA microarrays. So we can determine what genes are expressed under germ-free conditions. And then we compare this to the genes that are expressed under conventionally colonized conditions. So these are mice that have a normal, diverse microbiota in their guts. And so we look at the types of genes that are expressed under these conditions as well. And so if you compare these two DNA microarray experiments, you can come up with a list of microbiota-inducible genes. So these are genes that are essentially off in germ-free mice and are on in conventionally raised mice. And we initially reasoned that this list of microbiota-inducible genes might include genes encoding proteins that are important for defending the surface of the intestine against the trillions of bacteria that reside in the gut lumen. And so we, of course, came up with a long list of microbiota inducible genes, and we kind of sifted through these genes and looked for interesting hits. And one of the genes that has been especially interesting to us and we worked on for most of the last 8 or 10 years or so is a gene encoding a protein called RegIIIγ gamma. So RegIIIγ gamma is a member of a large family of proteins called the C-type lectin family. And most of the members of this family of proteins encode proteins that bind to carbohydrate ligands. And I'll tell you in a minute about the carbohydrate ligand that is bound by RegIIIγ gamma. So the name RegIIIγ gamma actually derives from the initial discovery of RegIIIγ gamma in pancreatic islets. So it was initially named regenerating islet-derived protein gamma for this example, and there's also an alpha and beta in mice. But its major site of expression is not the pancreas, as it turns out, it's the small intestine, and that's where we found it in our transcriptomics analysis. And you can see here that if we look at the messenger RNA levels for the gene encoding RegIIIγ gamma, it's practically undetectable in germ-free mice. So you don't see much of this gene expressed under germ-free conditions. But if you have bacteria present in the gut, so this is these are called conventionally raised mice, you see quite a bit of RegIIIγ gamma expression. And we and others have solved the crystal structure for the human form, the human equivalent of RegIIIγ gamma, which is actually confusingly named RegIIIα alpha. So this is the crystal structure, and it's got a structure that's very typical for a C-type lectin. Another point that I'll make is that this is a very small protein that is secreted from the surface of the intestinal lining, so it's secreted off of the surface of epithelial cells and into the lumen of the gut where you might expect that it's going to encounter bacteria. And that was what really motivated the next experiment that I'll tell you about, where we were trying to find out what this protein's function was. So we knew that it was expressed, prominently induced in response to the microbiota, but we didn't know anything about its function. And so one of the many experiments that we did was to make large quantities of RegIIIγ gamma in cells, purify this protein, and then add it to different bacterial cells in test tubes, essentially, and then see who survived. So this is called a dilution plating assay. So on the y-axis, you have the number of bacteria that are remaining after a 2-hour exposure to RegIIIγ gamma in a test tube. And so what you can see is that if we look at bacteria such as Listeria monocytogenes, Enterococcus faecalis, or Staphylococcus aureus, all of which are gram-positive bacteria, and I'll explain on the next slide the difference between a gram-positive and a gram-negative. What you can see is that these bacteria don't fare too well. They don't survive, and this dilution plating assay tells us that RegIIIγ gamma can kill these bacteria. In contrast, gram-negative bacteria such as E. coli are fairly resistant to the effects of RegIIIγ gamma. So this was very interesting to us because it told us that RegIIIγ gamma is a bactericidal protein, and it specifically kills gram-positive bacteria. And one of the reasons this was interesting was because this was the first example of a C-type lectin that could do this. So one of the first clues we got about how RegIIIγ gamma identifies and kills its bacterial targets had to do with this difference between gram-positive and gram-negative bacteria. So this was an essential clue. And I'm not going to show you the data for this, it's all published, but what we found is that RegIIIγ gamma actually binds an essential component of the bacterial cell wall called peptidoglycan. So peptidoglycan is a very long carbohydrate chain. It consists of N-acetylglucosamines and N-acetylmuramic acids. These are sugar molecules that are linked together in long chains. And these chains are linked together, the parallel chains in the bacterial cell wall are linked together by peptides that are part of MIRNAC. So what you end up with is a very rigid cell wall structure that is an excellent protective covering for the bacterial membrane. And this gets to the critical difference between gram-positive and gram-negative bacteria that was an essential clue for us. So in gram-positive bacteria, the peptidoglycan, which is in orange here, is generally speaking on the outer surface of the bacterial cell. What's different about gram-negative bacteria is that these bacteria have an outer membrane. So they have another membrane that covers and sort of protects this peptidoglycan component of the cell wall. So if RegIIIγ binds peptidoglycan, it's only going to be able to bind in the case of gram-positive bacteria. So this was one essential clue that gave us insight into how bacteria recognize and lock on to their bacterial targets in the gut. But how do they actually kill? How does a lectin actually kill a bacterium? So one clue that we got for this was in an experiment that we did where we incubated bacteria in the presence of two things. First of all, we added human RegIIIα, alpha, and the reason we used RegIIIα, alpha, the human form of essentially the human ortholog of mouse RegIIIγ gamma here, was because we had the crystal structure for this protein. And so we reasoned that we could relate what we learned about this protein in our biochemical experiments to the three-dimensional structure. So that was our rationale. But essentially, RegIIIα alpha is the same functionally as mouse RegIIIγ gamma. So we took bacteria, such as Listeria monocytogenes, and we incubated them in the presence of RegIIIα alpha and also in the presence of a membrane impermeant fluorescent dye called cytox. And so cytox is excluded from the interior of bacteria if there is an intact bacterial membrane. And so if bacteria take cytox up, it means that their membranes have been damaged. And you can see here from this graph, that's exactly what happens when you add RegIIIγ gamma to these bacteria in the presence of cytox. They take up RegIIIγ gamma over time. And that doesn't happen in the case of a control protein, which is bovine serum albumin here. So this told us that RegIIIα lectins disrupt bacterial membranes and gave us some insight into how they might actually be killing bacteria. We got some further insight into this by doing another experiment looking at the membrane disruptive activity of RegIIIa lectins. So this is called a dye leakage assay, and it's kind of the reverse of the assay that I just told you about in bacteria. So here we're making an artificial membrane. So this is an artificial membrane bilayer. And what we've done here is to encapsulate a fluorescent dye. So that here the dye starts out inside the membrane. And then we add RegIIIα alpha. And if it damages the, this is called a liposome, the circular structure. If it damages the liposome membrane, the dye will be released and we can measure that release. And so you can see here that when we add RegIIIα alpha to our dye encapsulating liposomes, we get rapid dye release. Whereas if we add just a buffer, we don't see a release unless we add detergent here at the end of the experiment. So this told us that RegIIIa lectins target membranes directly. So you don't need peptidoglycan to target the membranes. You need that to identify the bacterial targets, but then this protein can then damage the bacterial membrane. So this told us how these lectins can kill bacteria in the gut. We wanted to go a little deeper, though, and understand the mechanism by which this antibacterial molecule actually damages bacterial membranes. And one really fascinating insight we got into this was when we took our purified RegIIIα alpha protein, so remember RegIIIα alpha is the human form of RegIIIγ gamma, and we put it on artificial liposomes and then visualized these liposomes under electron microscopy. And so what you might be able to see here is that you wind up with these a lot of these fairly uniform circular structures. And on the right is a panel showing one of these structures in close-up view. So this is about 100 angstroms in diameter. So this is a really tiny particle. But what was really fascinating about this was that it's circular. It suggested that the protein is forming a pore on the membrane. And indeed, we were able to determine that these pores, using biochemical approaches that I'm not going to talk about here in detail, we were able to determine that the pores are RegIIIγ alpha hexamers. So there are 6 protein molecules per pore structure here. And so then we wanted to understand in much more detail how these pore structures were assembled, how they interacted with the membranes, in order to gain some additional mechanistic insight into this pore assembly on the bacterial membrane. And so to do this, we used an approach called cryo-electron microscopy. And so cryo-electron microscopy is an approach that people use in order to determine the structures of macromolecules. So if you have a large protein complex and you can visualize it, as we did, you can assemble hundreds of thousands of these images and get a much more high resolution picture of how these protein complexes are assembled. So unfortunately for us, the RegIIIγ alpha hexameric pore structure was actually too small to perform this technique. But what we noticed is that if we incubated RegIIIγ alpha for a long period of time with these artificial membranes, over time you essentially got what we interpreted to be a stacking of these pore subunits. And this created a very large macromolecular complex that we could then use to solve the structure. We were able to solve the structure by cryo-EM, and this was done in collaboration with Dr. Qiuxing Zhang in the Department of Cell Biology at UT Southwestern. He's now at the University of Florida. And so what Dr. Zhang did was to solve the RegIIIγ alpha filament structure, and then computationally extract the structure of the RegIIIγ alpha pore subunit. And it looked kind of like what we would predict based on what we saw under the microscope. Its diameter was about 90 angstroms, which was similar to the diameter of the pore that we saw under the microscope. And it was about 55 angstroms in height. And what's interesting about this is that this is just about exactly the height of a typical membrane. So this was sufficient. You could think about this as penetrating through a bacterial membrane. So I'm not going to go into a lot of these details, but we were able to take our RegIIIγ alpha crystal structure, dock it into our cryo-EM map, and gain some additional information about how these RegIIIγ alpha subunits interacted with each other. Furthermore, this allowed us to understand how this pore complex was stabilized in the membrane. So how does a protein that's in aqueous solution partition into a membrane and create a pore? So again, I'm not going to go into the details, but we were able to understand this using this technique. So, and it has to do with how the positively charged amino acids on the pore interact with negatively charged lipid head groups on the bacterial membrane. So now we have a fairly detailed model of how a C-type lectin can kill a bacteria. So you have either RegIIIγ gamma or RegIIIγ alpha, which are again functionally virtually identical. These proteins bind to peptidoglycan, which is on the outer surface of the gram-positive cell surface. And then in a process we don't understand very well yet, these lectins penetrate through the thick peptidoglycan layer, and we actually think that this happens during cell division. So when bacteria divide, there's a little gap in their peptidoglycan that has to be repaired. And so we think that RegIIIγ alpha slips in this gap during cell division. Ultimately, it comes into contact with the bacterial membrane, which is the very thing being protected by the peptidoglycan, and forms a pore, a hexameric pore. So essentially it punches a hole in the bacterial membrane. And so you wind up getting osmotic lysis, leakage of cytoplasmic components. All of these things are very bad for the bacteria, and the bacteria usually end up lysing and dying. So that's what we know about the biochemical function of RegIIIγ alpha and RegIIIγ gamma. So these are secreted from the surface of the gut and come into contact with bacteria and kill them through this mechanism. But what are they actually doing in vivo? What kinds of bacteria are they killing? Where are they killing them? And how does this impact mammalian host physiology? So that was another question that we wanted to answer in the past several years. And so before I tell you what we've found out about this, I want to return to the surface of the intestine and tell you a little bit more about the physical barrier that exists between you and your microbiota. So I told you before, and again, I'll refer to this very nice illustration of the small intestine, that there's a single layer of epithelial cells between you and your bacteria. There's actually another component to this barrier that's very important. In addition to the physical barrier presented by the epithelial cells, there's also a mucus layer. So the entire surface of the intestine is covered with a fairly thick mucus layer. It's about 150 microns thick. And this mucus layer essentially acts like flypaper. And I'll show you an actual microscopy image of this on the next slide. But to summarize what I'm about to tell you, basically the bacteria that are in the lumen of the gut tend to get stuck in the outer regions of this mucus layer, and this prevents them from accessing the surface of the intestinal epithelium. And as you might imagine, this is a very important aspect of how we protect ourselves against invasion by this vast microbiota. So what I'm going to tell you about RegIIIγ's role in all of this is that it's very important for keeping this inner mucus layer fairly free of bacteria. So it's kind of like a little landmine, which is part of how we keep bacteria confined to the outer mucus layer and to the lumen of the gut. So our insights about RegIIIγ's role in defending the inner mucus layer were actually preceded by some basic insights into how bacteria are spatially organized in the intestine. And so the way that we looked at this was to first try to visualize spatial relationships between the microbiota and the epithelial surface. So where are the microorganisms relative to our tissues, and what happens when you take RegIIIγ out of the equation? So we did these studies using a technique called fluorescence in situ hybridization, or FISH. And what this involves is the use of a DNA probe, which is linked to a fluorescent molecule, so we can visualize it under a microscope. And we can hybridize this probe to a bacterial gene. And we use the 16S ribosomal RNA gene because it's only, this particular gene sequence is only in bacteria, and you don't have it in host cells, so you can specifically identify your bacteria. And so if we take this probe and we layer it onto a slice through a mouse small intestinal tissue, which is shown here, further taking care to preserve the tissue so that the mucus layer is also preserved, and it doesn't always happen depending on what fixation techniques you use. You can see that the bacteria are sort of kept at arm's length. So this is the mouse tissue down here. So this is the small intestinal cells, and you can see the epithelial surface here. This blue stain is a nuclear counterstain, and your green bacteria are out here. So there's about a gap of about 50 microns between you and your bacteria at the intestinal surface. And just to give this thing a name, this name is actually stuck. We call it the demilitarized zone, or the DMZ. So there's this gap between you and your bacteria that is likely very important for promoting the symbiotic nature of this relationship. So what we wanted to know was, how is this DMZ maintained? Is it just a function of the physical properties of the mucus layer, or is the immune system and the proteins, are there proteins made by epithelial cells that might be important for maintaining this essential gap between bacteria and the intestinal surface? And so one of the clues that we got about this was obtained when we looked at some mice that we had in our colony. So these were mice that were genetically engineered to lack a critical component of the immune system. So this component of the immune system is MyD88. So this is an essential signaling molecule that's involved in signaling through toll-like receptors. So toll-like receptors are a very important component of the innate immune system in all mammals. And so what these receptors do is they detect things that are made specifically by bacteria. We call these microbial molecular patterns. In the case of bacteria, these are things like lipopolysaccharide, flagellin, and so forth. These are molecules that are only made by bacteria and are never made by mammalian cells. So if a cell detects these molecules, it means that there's trouble. So toll-like receptors detect microbial molecular patterns, and MyD88 is critical for signaling this sensing to the nucleus, where it upregulates the expression of things like antimicrobial proteins and cytokines that are important for the immune response against microorganisms. And so what was interesting was that when we looked at mice that are engineered to lack MyD88, so they can't signal through their toll-like receptors, we saw that they don't have a DMZ. So when we did this FISH technique, we saw a lot of bacteria sort of crowding up against the intestinal surface, and this wasn't true in mice that were wild type and maintained under the same conditions. So this told us an important clue. So the immune system is indeed involved in this. MyD88 and toll-like receptor signaling are important for limiting bacterial contact with the epithelial surface. And at the same time that we had this insight, we had another insight into what controls RegIIIγ gamma expression in the gut. And so what we found was that when we deleted MyD88 from intestinal epithelial cells, we lost RegIIIγ gamma expression. So you can see here when we stain with antibodies for RegIIIγ gamma in wild-type mice, so these are mice that harbor what we call a floxed MyD88 allele, and you can just think of these as being basically wild-type mice. So they express RegIIIγ gamma, that's shown in red here. But if you delete MyD88 from the epithelial cells, RegIIIγ gamma goes away. So that told us that not only is MyD88 necessary for bacterial association with the epithelial surface, but it also controls RegIIIγ gamma expression. So it does these two things. And so that led us to the question of whether RegIIIγ gamma might be the MyD88, part of the reason why MyD88 is important for limiting bacterial association with the epithelial surface. So we addressed this question directly by engineering mice to lack RegIIIγ gamma. So we deleted the RegIIIγ gamma gene from mice and then looked at whether they had a demilitarized zone or not, and you can see that they don't. So if we look at RegIIIγ gamma knockout mice, and here we're looking at a wild-type litter mate. So this is a litter mate that is living in the same cage as the RegIIIγ gamma knockout mice, and so it's seeing this, it's got essentially the same microbiota. You can see that there are, again, a lot of bacteria crowded up against the intestinal epithelial surface. So because we know from our biochemistry that RegIIIγ gamma specifically targets gram-positive bacteria, that led us to investigate a kind of a nuanced question here, which was we would predict that these bacteria that are sort of colonizing the epithelial surface in these RegIIIγ gamma knockout mice would be more likely to be gram-positives. So does RegIIIγ gamma preferentially limit surface association of gram-positive bacteria? And that indeed turns out to be the case. So when we scraped off the bacteria from the intestinal surface of RegIIIγ gamma knockout mice and compared them to wild-type mice and did a technique called 16S ribosomal DNA sequencing. Basically, this is a culture-independent technique that I referred to in my first lecture. We find that there are more firmicutes represented as compared to Bacteroidetes. So Bacteroidetes are a gram-negative phylum, firmicutes are a gram-positive phylum. So you have increased representation of the firmicutes or some firmicutes in the RegIIIγ gamma knockout mice. So what we think we've identified here is a very interesting negative regulatory feedback loop at the intestinal surface that involves sensing of microbe-associated molecular patterns by toll-like receptors, which signal through MyD88 and then turn on RegIIIγ gamma expression. RegIIIγ gamma, the RegIIIγ gamma gene makes the RegIIIγ gamma protein, which is then secreted into this demilitarized zone and helps patrol this zone and keep bacteria from contacting the epithelial surface. And as you might imagine, just keeping bacteria from attaching to the surface of the intestine is solves 90% of the problem of how to keep them out of the tissues. And so we think that this is a very important element of how we how we maintain a symbiotic relationship with the vast numbers of bacteria that colonize the intestine. And we're now trying to determine, so RegIIIγ sort of tells us how we deal with gram-positive bacteria in the gut. But the question of how we deal with gram-negatives, how do we keep those out of our tissues, is still remains to be answered, and that's something that we're working on actively now. So I'll stop there, and I want to, because this lecture was about work from my lab, I want to give a special thanks to all of the many students and postdocs that have been in my lab over the past 10 years or so. And in particular, I want to point out two individuals. Dr. Sohini Mukherjee did all of the biochemistry on RegIIIα alpha that I talked about, and Dr. Shipra Vaishnava did the in vivo work on RegIIIγ gamma and the demilitarized zone. Of course, I also would like to thank the agencies that fund our research, including the National Institutes of Health, the Howard Hughes Medical Institute, and the Burroughs Wellcome Foundation. Thank you.